Welcome to my channel Deep Learning Biology. Now in this video we are going to solve more numerical problems related to DNA RNA quantification. Now hopefully you have watched the video number 1 and video number 2. Okay, now this is the third video, okay, related to DNA RNA quantification. So here hopefully you remember, for double stranded DNA, okay, for double stranded DNA 1 OD, it is going to express 50 microgram DNA per ml. Okay, for single stranded DNA, okay, 1 OD, okay, it is going to represent 33 microgram per ml, and for single stranded RNA, okay, it is 40 microgram per ml. Okay, now using this information you can easily calculate the concentration of DNA, whether it is double stranded DNA, single stranded DNA, of RNA if it is single stranded RNA. Now try to see here, let us try to solve one numerical problem. Okay, 40 microliter of a stock solution of RNA is diluted with water to give a final volume of 1000 microliter. The diluted sample has an absorbance, okay, absorbance 260 nanometer, 0.142, okay, 0.142. What is concentration of RNA of stock? Okay, now hopefully you remember, okay, for single stranded RNA, okay, 1 OD is equal to 40 microgram per ml. Okay, now to solve this, try to see here, you are having a stock 40 microliters stock solution and you diluted it with a final volume 1000 microliter. Okay, so you are having 40 microliters and you make the final volume 1000. What is going to be the dilution factor? 1000, okay, divided by 40. The diluted sample has an absorbance 260, 0.142. What is the concentration of RNA of stock? Very simple now. Okay, concentration of RNA, it will be absorbance at 260 multiplied by dilution factor multiplied by this one 40 microgram per ml, conversion factor. Okay, so here concentration of RNA will be, what is the value of absorbance at 260? 0.142. What is the dilution factor here? Final volume 1000, initial volume 40, okay, and you have to multiply it with 40 microgram per ml. If you are going to solve this, it will come out 142.5 microgram RNA per ml. Okay, yes, you can also expressed per microliter. Just convert ml into microliter. Okay, so it will be 142.5 microgram per 1000 microliter, and if you are going to solve this, it will be 0.142 microgram per microliter. Okay, so hopefully you have learned. These are very quite simple questions. Okay, now let us try to solve another simple question. Okay, how much DNA is present in a single diploid cell? How much DNA is present in a single diploid cell? Okay, now try to see here. If you see, okay, here we are talking about human cell. Okay, and human diploid cell. Okay, if you see within the human, okay, if you see the genome content, total DNA content, okay, it is being expressed 3 into 10 to the power 9 base pairs. So generally, if you see the total DNA content which is present within the human diploid cell, we are going to have 3 billion letters. But here I just want to clarify, this is haploid DNA content. This is haploid DNA content. Okay, and here we have to see what is going to be the total DNA in a single diploid cell. Okay, so what will be the weight of that DNA? Okay, so here within the human genome, you are going to have 3 billion letters, 3 billion base pairs. So it is going to express the haploid DNA content. So for diploid cell, okay, keep in mind it will be 2 into 3 into 10 to the power 9 base pair. So if you calculate, it will be 6 into 10 to the power 9 base pair. So DNA which is present within the diploid cell, it is 6 into 10 to the power 9 base pair. Now what is going to be the weight of this DNA? How much it is going to weigh? Yes, you can calculate easily. Okay, just recall, hopefully you remember, for double stranded DNA, okay, if you see the molecular weight, it is 660 gram per mole. This you have to remember. For double stranded DNA, the molecular weight is 660 gram per mole. Means for double stranded DNA, 1 mole, it is going to weigh 660 gram. And hopefully you remember, 1 mole, it is going to represent 6.023 into 10 to the power 23 molecules. Okay, 6.023 into 10 to the power 23 molecules, they are going to weigh 660 gram. Now DNA is a polymer. So here the each nucleotide, they have been joined together, and here if you weigh one nucleotide and if you sum the weight of each nucleotide, it is going to tell you the weight of the DNA. Try to see here how many base pairs we are going to have here. Okay, so here you see these molecules, okay, you can consider as one base pair. Okay, here one base pair, it is going to weigh, okay, 660 divided by 6.023 into 10 to the power 23. Try to see here, if you see the 4 double stranded DNA molecular weight is 660 gram per mole. 1 mole is going to weigh 660 gram. Okay, 6.023 into 10 to the power 23 molecules. okay, of A pair with T, G pair with C. Okay, if you are going to have these base pairs, okay, and if you are going to have this 6.023 into 10 to the power 23, they are going to weigh 660 gram. Then what will be the weight of one base pair? 660 divided by 6.023 into 10 to the power 23. Now if you want to calculate what is going to be the weight of 6 into 10 to the power 9 base pairs, very simple. Okay, what is the weight of 1? Okay, it is 660 divided by 6.023 into 10 to the power 23. Okay, this is the weight of one base pair. And what will be the weight of 6 into 10 to the power 9 base pair? Just multiplied by this. Very simple. Okay, now here it will come in gram. Okay, if you want to convert this into picogram, it's simple. 660 into 6 into 10 to the power 9 gram, you can convert into picogram. Just multiply it with 10 to the power 12. Your answer will come in picogram divided by 6.023 into 10 to the power 23. And if you are going to solve this, it will come out 7 picogram. So keep in mind, the weight of DNA, the total weight of DNA in human diploid cell is 7 picogram. Hopefully you have understood this. Okay, please do like, share the video and subscribe to my channel Deep Learning Biology. Bye bye, take care.